Hey everyone, welcome. Today we're going on a truly cinematic journey into the microscopic world of the human body, where we reveal the invisible architecture of life. In this explainer, we're going to explore the fascinating chemical tools that allow us to see what would otherwise be completely hidden from the human eye.
You know, if you just put human tissue under a microscope, it's actually completely transparent. It's literally like looking at a clear pane of glass. But with the right chemical key, each stain reveals a hidden story inside that tissue. Suddenly, we can uncover specific microorganisms, connective tissue fibers, and abnormal protein deposits that are otherwise impossible to detect. So in today's explainer, here's our game plan. We'll be covering the histopathology gold standard, the chemistry of alum hematoxylin, special stains in pathology, beware the formalin pigment, fixing for electron microscopy, and finally, light versus electron microscopy. Let's get right into it.
Starting things off with Section 1, the histopathology gold standard.
We've got to start with Hematoxylin Eosin, or H&E as everyone calls it. This is the universal default standard. It's performed millions of times a day in labs all around the world. It's the essential baseline that provides the necessary contrast to tissues and cells, allowing pathologists to make critical diagnoses.
To put that into perspective, take a look at this 5-micron section of renal cell carcinoma. The contrast here is just stunning, right? Hematoxylin specifically targets the nucleus. It acts as a basic dye that paints basophilic structures like nucleic acids a deep blue-purple hue. Meanwhile, the eosin acts as an acidic countersting, coloring the eosinophilic structures, things like the cytoplasm and extracellular matrix, in varying shades of pink. It's basically a beautiful, highly functional map of the tissue.
Moving on to Section 2, the chemistry of alum hematoxylin.
Hematoxylin actually comes from the logwood tree, and it requires a very specific chemical grip to work. We generally approach this staining in two primary ways. First up is progressive staining. This is kind of a gentle approach, using solutions like Mayer's hematoxylin, where we slowly bring out the nucleus until it turns a deeper blue. Alternatively, we use regressive staining, like with Harris hematoxylin. This is a much stronger approach, where we deliberately overstain everything and then use a dilute acid to wash it back, or differentiate it, until we achieve that perfect, crisp, nuclear contrast.
What's really fascinating is the precise chemistry behind that grip. If you look at how different alum hematoxylin stack up, Harris hematoxylin is a clear winner for firmness. Why? Because it has a high mordant content and a low dye content. A mordant is basically the chemical bridge that attaches the dye to the tissue. Tons of mordant means it binds super firmly, resulting in amazing enhanced nuclear staining. In contrast, Mayer's and Ehrlich's have higher dye content but lower mordant, meaning they bind much more loosely.
All right, let's jump to Section 3, special stains in pathology.
While H&E is fantastic for the big picture, some cellular elements remain completely invisible. That's where specialized chemical tools step in. Imagine smoothly zooming right down into the tissue in stunning 3D detail. If pathologists are hunting for acid-fast bacteria, they use the Ziehl Neelsen stain, which makes tuberculosis bacilli glow bright red against a blue background. Or take PAS, periodic Acid-Schiff. Hit tissue with this, and fungal hyphae or mucin suddenly pop in a brilliant, vivid magenta. And if we're looking for hidden iron deposits, Perls Prussian blue turns hemosiderin inside macrophages a striking deep blue. Every special stain reveals a totally distinct microscopic target.
Next up is Section 4, beware the formalin pigment.
Okay, picture this. You've prepared the perfect paper-thin tissue section. You've hit it with these brilliant special stains, and you find the microscopic details are completely obscured by a dark, ugly smudge. What chemical reaction causes this absolute diagnostic nightmare?
The villain here is formalin pigment, technically known as acid formaldehyde hematin. If you make the mistake of using unbuffered formalin to fix blood-rich tissues, the acidic environment allows the formaldehyde to react directly with hemoglobin, and boom, you get this dark brown to black granular artifact that deposits right over your tissue, completely ruining the delicate histological details we worked so hard to preserve. And hey, here's a wild little lab fact that often catches people off guard. A standard 10% formalin solution, which is pretty much the cornerstone fixative in virtually every lab, actually only contains exactly 4% dissolved formaldehyde gas. It's crazy, but in this field, precise chemical ratios are absolutely everything.
Let's push even further. Section 5, fixing for electron microscopy.
What if we want to zoom past the limits of light microscopy, right into the ultrastructure of the cell itself? Well, standard formalin simply won't survive the intense, high-vacuum environment of an electron microscope. You need extreme stabilization. Step 1 requires glutaraldehyde. Because it's a dialdehyde, it rapidly cross-links proteins and beautifully preserves ground substances like glycogen. But glutaraldehyde leaves lipids completely unprotected. So step 2 is mandatory, a secondary fixation using osmium tetroxide. This reacts heavily with lipids to lock them into place so they aren't extracted during dehydration.
Which brings us to the cinematic climax of our microscopic journey. Section 6, Light versus Electron Microscopy.
Comparing these two views of skeletal muscle is honestly mind-blowing. It's like comparing a broad map of a city to standing right in the middle of a bustling street. The top view, from a light microscope, gives us the macro layout. We see the muscle fibers and the general location of a blood vessel. But that bottom view, that's the electron microscope. The level of intricate ultrastructure is stunning. You can literally see the actual banding patterns of the muscle sarcomeres and the dense mitochondria packed right between them.
But here is the massive catch. Your chemical preparation technique dictates exactly what you can see. Look what happens to an electron micrograph when the tissue is only fixed with standard formalin and paraffin. It's an absolute disaster. Crucial cellular components, especially lipids and fine protein structures, have simply washed away during processing. We are left with these gaping holes and degraded nuclei. You are literally missing half the story.
Now check out the difference when it's done right. This tissue is perfectly fixed using that two-step glutaraldehyde and osmium tetroxide method. The cellular architecture is preserved with breathtaking clarity. The nucleus is totally intact with sharp chromatin details, and those large, smooth lipid droplets are locked perfectly in place, exactly where they belong. It's literally night and day.
So, whether we're relying on the basic, brilliant contrast of H&E or diving into the ultra-high-resolution depths of the electron microscope, choosing the correct chemical tool is the ultimate key in diagnostic histopathology. We aren't just passively looking at tissues. We are actively engineering them with chemistry to reveal their deepest secrets.
Which leaves us with a pretty massive question. As we continuously refine these microscopic visual guides, and as technology leaps forward with 3D digital pathology and advanced molecular labeling, what other hidden stories inside our tissues are still waiting to be illuminated? Will the chemical and digital stains of tomorrow make our current microscopes look like simple magnifying glasses? Definitely something to think about next time you look at a medical image. Thanks for exploring the invisible with us on this explainer.